Hey guys. Um, so, this is a lecture about foodborne illness. I've spent much of my career in food safety regulation, foodborne illness prevention policy, and foodborne illness outbreak investigation. So, the subject of foodborne pathogens and disease is near and dear to my heart. Foodborne illness epidemiology is one of the most vibrant and gratifying areas of public health. Also, people are often familiar with the concept of a foodborne illness outbreak. So, this can serve as a great way to explain applied epidemiology and public health. Thankfully, many foodborne illnesses are mild or self limiting, meaning people get better just on their own without treatment. But occasionally they can get nasty, requiring medical treatment or hospitalization, or even be deadly. Symptoms usually are gastrointestinal, like vomiting and diarrhea, but some bugs can cause severe consequences like organ failure or hemolytic uremic syndrome, or unfortunate sequelae like arthritis or a neurologic syndrome called Guillain-Barré. Food safety and foodborne illnesses are key aspects of One Health. Many foodborne pathogens that make people sick are from animals, and these can transmit to people through food, direct contact with animals, or both. Other foodborne pathogens have reservoirs in the environment, such as recreational water or drains in food processing facilities. So, foodborne illness again, it really connects the One Health triad. So, this lecture is intended to give you just some brief concepts. Many of you have learned about foodborne illness and for sure about epidemiology concepts in other courses. This is just an introduction to this concept, and hopefully will be the foundation that you can use as a springboard to further learning about it. Just some basic concepts. So this page here that you're seeing is just to give you an idea of the illness burden in the United States. So CDC estimates that 48 million people get sick each year, 128,000 are hospitalized, and 3,000 die from foodborne diseases each year in the United States. I will be providing links to all of these resources in the module. So it's a big problem. Foodborne illnesses are a major problem. Many people experience them. Thankfully, most are just mild. But even the mild ones, nobody likes to get sick. We don't want to get sick from our food. So it's a really good thing to pay attention to, do surveillance for, investigate, respond, prevent, and prepare for. So the FDA has some great information about foodborne illnesses as well. Kind of some of the similar information right here. Those are based on the same study. What you need to know about foodborne illnesses. This is a really, really helpful chart that the FDA has created. And it's a table of foodborne illness causing organisms. You've got organism, onset time after ingesting, in other words, incubation period. Got signs and symptoms, duration, and food sources. So here are listing a bunch of the organisms. You can see foodborne pathogens. Generally, they're bacteria, viruses, or parasites. Almost anything can be a foodborne pathogen, but these are like the main ones. And even on this list, some of these are more rare or occur less often, or are just less of a problem, make people only a little bit sick. So I want to show you a few of them here, just highlight a few things about this. So see this here, onset time after ingesting. If you see things that are 10 to 16 hours, very like less than a day to onset, between the time of consumption and the time of symptoms start, that is likely going to be a foodborne intoxication versus illness. And here's another resource, the FDA Bad Bug book. You can read much more in depth about all these things, but I'm going to find, okay, where is it here? Yeah, right here. Food intoxication, a form of food poisoning caused by the ingestion of microbial toxins produced in foods prior to consumption. So certain bacteria will make a toxin and it'll just be in the food. So you don't even have to have the bacteria present there. In this case, people are eating the toxin, and it's the toxin that makes people sick. So in other words, intoxication. Then you have foodborne infection, which is a different type of thing, which is actually the food actually contains these pathogens, and then the pathogens cause an infection inside a person and either directly wreak havoc, or they may create toxins while they're inside a person. So there's two key concepts, foodborne infection and foodborne intoxication. Either one will cause foodborne illness, but since the toxin is already formed in foodborne intoxication, that's why you see some of these which are less than a day between the time of eating and the time of symptom onset. So Bacillus cereus, Clostridium perfringens, and down here Staphylococcus aureus are great examples of organisms that cause foodborne intoxication. Here's staph, you get a 1 to 6 hour incubation period. When you start getting things that go beyond a day, as far as incubation period, those are more likely foodborne infections. Mixed up here, I really wish on this table they had put which type of pathogen it is, whether it's a bacteria, virus, or parasite. But just know that there's multiple different types of foodborne pathogens. Okay, again, this Bad Bug book, which is a great title, you can look this up, Google it, if you really want to dive into more of the details on each of these pathogens. It's the Handbook of Foodborne Pathogenic Microorganisms and Natural Toxins. It's a great reference from the FDA. Okay, now I want to talk about vehicle. Let's see. Where is it? Here. Okay. Novel outbreak associated food vehicles, United States. So I want to just introduce this term of vehicle. In the previous module, vector-borne diseases, we talked about vectors that pass infections, pathogens between animals or potentially an environmental reservoir and people or other hosts, like mosquitoes and ticks. That's a vector. When we talk about foodborne illness, we use the term vehicle, a food vehicle, or even a water vehicle. It's the same idea, just some sort of intermediary that transfers the pathogen to an infected person or animal. Food vehicle. And anything can be a vehicle. Here's an article about novel vehicles for foodborne outbreaks. So if you wanted to look at those, but a common one is, say, ground beef gets contaminated with E. coli, it's undercooked, somebody eats it, and then they get sick. There's lots of others. Lettuce, spinach, raw milk, those are all examples of food vehicles. I want to talk, this is mainly about foodborne infections and foodborne illnesses, but Water can be a great, a really efficient, effective vehicle. Contaminated water can make a lot of people sick. And I want to introduce John Snow. He's considered the father of modern epidemiology. There was a cholera epidemic in 1854 in London, and he figured out that that water from the Broad Street pump was making people sick. And here, I guess I can't zoom in on this anymore, but here you can see this is a map of a part of London, and all these black dots. He took this map and he like filled in, and the higher the dashes go, this column of dashes go from the street, the more cases of cholera infection there were. So you can see kind of through here they were clustered, and if you were able to zoom in, you'd see that this right here is Broad Street, and there's a pump right here, and the famous Broad Street pump. He provided the evidence for the authorities to remove the handle of the Broad Street pump and stop the outbreak. That the water in the Broad Street pump was contaminated with cholera. So these types of methods, trying to figure out where people were getting sick, who are they, in what time period, and then what exposures they had, were really the foundation of some of our modern epidemiologic principles. And so we celebrate John Snow as important in the history of epidemiology. Well, this was a great waterborne infection. When we talk about foodborne infections, we're talking also about water, and basically anything that's consumed. Beyond that, we also can talk about enteric diseases more broadly. So not just foodborne, or waterborne, or anything that's consumed, but also things like direct contact with animals transmitting pathogens that give people gastrointestinal or enteric illness. Often these are thought of as a category together. Salmonella, for example, can contaminate food and people eat it and get sick, but also people can get salmonellosis from directly touching animals. Say they touch a chicken and it gets some salmonella on their hands, or bearded dragon or something. They can get dirty hands, and then maybe they don't wash their hands well enough before they eat next, or perhaps they prepare food with dirty hands and feed it to others. Those are all ways, various ways that people can get sick. It almost doesn't matter too much the particular vehicle when you're thinking about just pathogens in general. So we often think of enteric diseases, regardless of the source, regardless of the vehicle, as a category of epidemiology, if you will. Also, there are pathogens that don't come from animals. Many do. Many foodborne pathogens come from animals. However, Listeria is an example of one that does not. It can infect animals and it can be in the animal environment, but really what causes people to get sick from foodborne Listeria is often when that bacteria sets up shop in a food processing facility, like in a drain, and then somebody sprays the drain with water and it splatters everywhere onto the food or food contact surfaces. That's not really a zoonotic, but it's definitely One Health, and it's a serious foodborne infection. That's just one example. Norovirus is another great example. And you can find some of these on this FDA list. So here's Listeria. Norovirus is right under it. It's not zoonotic, but it can hang out in the environment and pass from person to person. It can be foodborne. So there are, of course, non-zoonotic foodborne illnesses, but it's just a real mix. And for the purposes of One Health, we don't worry too much when taken as a whole category. Are they zoonotic? Are they not? Are they Is it directly from an animal? Is it directly from the environment? Is it through a food or waterborne vehicle? Taken together, it's all just enteric disease. Okay, I wanted to give you an idea about food regulation because these are the agencies. This is on foodsafety.gov. There's really three main agencies that regulate food at the federal level and that get involved in foodborne outbreak response, investigation, and prevention. The USDA Food Safety and Inspection Service, or FSIS, regulates meat, poultry, and egg products. And then the FDA regulates basically everything else when it comes to food, as well as other stuff like human and veterinary drugs, for example, pharmaceuticals. So between these two agencies, FSIS and FDA, basically they regulate all of the food that we eat in the United States. And then you have the CDC, that they take the lead on investigating and doing surveillance for foodborne illnesses. And these three agencies really collaborate for national foodborne outbreak response. And then there's state agencies, state public health and state agriculture. And there's a few other examples of state level agencies that get involved in food safety. They're really important players. And then the local level as well. Local health departments often get involved in foodborne epidemiology. So that's important to know the lay of the land from a regulatory and government player standpoint. As far as surveillance, there's a whole bunch of different systems for foodborne or enteric disease, more generally enteric disease surveillance. Here's a CDC page about the surveillance systems. And this page here kind of has a breakdown of foodborne illness surveillance systems. So FoodNet is one that tracks individual illnesses from food. There's a collection of states that partner to do this. So you're kind of looking at a selection of states submitting sporadic illness information. When I say sporadic, I mean non-outbreak or not part of a recognized outbreak. So you get some individual case information and it tracks trends of illnesses. NARMS has to do with antimicrobial resistance, which we'll get into in a future module. CaliciNet is for norovirus. You'll notice that it's kind of trendy to name your food surveillance system or even other some sort of net. And FoodNet and PulseNet were some of the originals. There's PulseNet. PulseNet was named because of the technology pulsed field gel electrophoresis. They used to take bacterial DNA, amplify it, cut it up with enzymes, and then run it on a gel in an electric current. And different size pieces of the DNA would settle out on the gel at different places. And then the pattern that created was like a PFGE or pulse field gel electrophoresis fingerprint. And that was a keystone, a key component of molecular epidemiology, a DNA fingerprint, if you will. Now we do whole genome sequencing, and it's like full or part sequencing of the bacterial genome. And it's the same idea, though. You're looking at a DNA fingerprint. It does provide quite a bit more specificity than PFG, but it's the same idea. The surveillance system is still called PulseNet. A little historical nod to PFGE, even though it's based now on whole genome sequencing or WGS. Then we have NNDS. We won't worry about that. NORS, this is the National Outbreak Reporting System. These are enteric disease outbreaks or foodborne outbreaks. So whereas FoodNet is a case based, individual patient illness based system. NORS is an outbreak based system. So what gets reported there is not people, but outbreaks of illness. And then there's environmental health surveillance is SNET. And there are others. This is just an example or some examples for you to be aware of. Okay. Finally, I want to, let's see, where is my, no, that's, here we go. Okay. I wanted to show you this, which is put out by NEHA, the National Environmental Health Association, Foodborne Illness Glossary. There's two main types of epidemiologic studies or tools that we use in foodborne epidemiology. The case control study, which is right here, and the cohort study. Case control studies when you have when you do not know the entire population exposed, but you do know some of the cases, and you can find some reasonable controls. So you're looking at the cases, ill people, and what they were exposed to compared to well people, not sick people, and what they were exposed to. And generally what you do with that is put it in a 2x2 table where you have this matrix of exposed or not exposed and ill and not ill. And then you calculate an odds ratio from this. And it can be statistically significant or not. And here's the odds ratio from this 2x2 table. I know you get that in your epi course, but just wanted to review it here in the context of foodborne epi and One Health. And the other one is a cohort study when you do know the entire population exposed. And this is a great thing for like a church potluck or something, or a restaurant. Maybe you can get a hold of the entire list of everybody who was there. And you know everyone who was exposed or potentially exposed to the food that's making people sick. And then you calculate, you figure out which foods everyone was exposed to. And then you can calculate, instead of an odds ratio, you can calculate relative risk, sometimes called risk ratio. So I just wanted to make sure you're introduced or reminded of some of those terms, some of these resources, and kind of just the general universe of foodborne illness epidemiology.